I'm going to talk about genetics, cilia, and the link to congenital heart disease. And after all the talks this morning, some of this stuff is probably somewhat redundant, so I'm going to kind of move through it a little more quickly. But first of all, what causes heterotaxy and heterotaxy-associated congenital heart disease? The thought is that a significant percentage of it is genetic, but what is the real evidence for that? So first of all, there is increased recurrence of heterotaxy in siblings of other heterotaxy patients. And amongst the subtypes of congenital heart disease, the recurrence risk in heterotaxy is amongst the highest. There's also a notable increase in heterotaxy patients amongst consanguineous populations. So when a group of congenital heart disease patients from Turkey, where there's a higher incidence of consanguinity due to social acceptance of consanguinity, heterotaxy was one of the types of heart disease that came up at a much higher level than in, for instance, the US population. And beyond that, there's intense genetic heterogeneity, and that's already been hinted at. There are multiple different inheritance patterns: autosomal recessive, X-linked, and dominant. You can have de novo mechanisms. There is some evidence for maternal effects, and I'm not going to be talking about those specifically. Maternal early gestational diabetes has been linked to heterotaxy, and heterotaxy is one of the heart diseases that might be most contributed to by maternal diabetes. X-linked, as has already been alluded to in El Geddes' talk. Single gene recessive is probably the most common genetic mechanism, and then a combinatorial effect of defects in multiple members of a single biological pathway is likely a significant contributor that we have, as of today, not been able to assay particularly well. So the first evidence was the existence of extensive pedigrees of heterotaxy segregating in Mendelian manner. For instance, this is a large X-linked pedigree. The gene was not identified. This, I think, was published in the 1980s, but it shows multiple generations with multiple people affected by heterotaxy. And the other thing I want you to take home from all these pedigrees is that the heart disease within one pedigree is incredibly variable. And there are individuals in the pedigree who are obligate genetic carriers who are entirely normal. They have no situs abnormality, no respiratory abnormality, no cardiac abnormality. Here is a recessive pedigree. This one was the X-linked. The recessive pedigree is the older one from 1983. But just like the X-linked pedigree, it shows the significant variability in the cardiac phenotype, along with obligate carriers that are completely normal. There's also dominant pedigrees. This is the activin receptor 2B gene, and clearly here it is dominantly inherited. And again, there is variability of the final phenotype. So what this suggests is even within a single genetic defect, there is tremendous phenotypic heterogeneity, which makes it more difficult to really accurately dissect the genetic contribution to heterotaxy and the associated congenital heart diseases. And the first place where we really kind of noticed this is when we started studying mice with heterotaxy, and this is really what started my whole business in heterotaxy, congenital heart disease, et cetera, is to look at a mouse that can't tell its left side from its right side, seen on the left side of this screen. And if you look at the cardiac abnormalities of the mice, which have a variant in DNAH11, one of the dynein heavy chains, you can see that some of them have atrial septal defects, ventricular septal defects, pulmonary stenosis, transposition. And this is from, again, the 80s and 90s. And when you look at human heterotaxy patients, they pretty much mirror what we saw in the mice, the tremendous variability in the phenotype. And the way these mice with a single gene mutation are maintained, and they're still in our mouse room to date, is that about 3 out of 10 are completely normal. And those completely normal mice have no heart disease, no problems whatsoever. They breed and maintain the line. Everybody else dies. And in mice, congenital heart disease is not well tolerated for reasons that are not totally understood. The mice with heart disease are usually lost mid to late gestation and never born, or picked up by the mom shortly postnatally and dealt with. So the key thing from all of this is there are multiple different ways you can inherit laterality type defects. The phenotype amongst any given gene is incredibly variable. And now what compounds it, if you start looking at some of these pedigrees more carefully, is that isolated congenital heart disease, things that we've never thought of as part of the heterotaxy spectrum. For instance, detransposition of the great arteries is found within these pedigrees, both in the human pedigrees and in the mice. So I think what this has made us think about for the larger genetic studies is maybe we shouldn't just be looking at the patients that are classified as dextrocardia, right atrial isomerism, left atrial isomerism, but patients across a broader spectrum and call it laterality-associated congenital heart disease, which for the purpose of the genetic studies I'm going to talk about going forward is defined as detransposition, isolated L-transposition, clear-cut heterotaxy, and even some forms of atrioventricular canal defect. When you look at the genetics across large human cohorts, it's amongst this larger population of laterality-associated congenital heart defects that you start picking up threads of specific genes causing the problem. So genes that contribute to laterality spectrum congenital heart disease are required for left-right asymmetry determination because those are the events that establish your left-right axis. And in humans happens around 17 to 19 days of gestation, so it's very, very, very early during the pregnancy. Most women at that point don't even know they're pregnant, and this event is already happening. In mice, it's day 7.75, as I've learned the hard way, occurs somewhere around midnight in the mouse room. It's very evanescent. And the structure that does all this, the left-right organizer, lives for about 12 hours and does its most important things for about 3 or 4 hours during that time span, making work on it a little more challenging. So this is a very diagrammatic version of a left-right organizer that you've already seen before, but basically it is a teardrop-shaped structure at the anterior end of the primitive streak at the end of gastrulation. And it is far separate from the heart. So if you're thinking about the left-right organizer and the heart, the left-right organizer would be here, and the developing heart would be way up here. So you make a signal at the left-right organizer that requires cilia motility, so these guys drawn in green, they're moving. By moving, they then make a mechanical contact with another cilium, which is thought to be the ones that are on the edge here, on the yellow cells. But it's also possible that some of these ones in here are able to both sense and create a signal at the same time. That signal is then converted into some molecular changes, including degradation of an RNA called DAN5. And that is really the part of this process that we are 100% sure about. You need cilia, they need to move in a normal direction, and there need to be sensing cilia that can receive that signal. Then what happens after you degrade DAN5 is a little unclear, but you do need a pathway called nodal signaling, which spreads whatever information is generated by the left-right organizer across the embryo, which is then received by the heart to allow the heart to say, hey, I'm going this way or this way. And some work by Siegel and Mayak strongly suggests that some of those early signals are in the atrium and in the outflow tract, coming back to our question about detransposition of the great arteries being a part of this spectrum. So what else do we know for sure about these early steps is that moving and the cilia and sensing that cilia signal is absolutely determinative of heart looping direction. So there was a guy who's now here in Boston who was in my lab to start out with, who did a really unique experiment where he was able to develop an optical tweezer system that grabbed a cilium in a zebrafish left-right organizer. And basically, the thing is not moving because it's paralyzed. Then he grabs it with the optical tweezers and wiggles it. You wiggle it for a certain amount of time, then you wait and see what happens to the heart. And if the cilium you wiggle is on the left side, the heart will always loop normally. Now remember, these guys have paralyzed cilia, so now what we're going to do, is we're going to do the same experiment on the right side. Take a cilium on the right side, wiggle it, and you guessed it, heart's going the other way. So this part of the pathway we are 100% sure about. You move the cilium, you determine the direction of the heart loop. But how that's propagated, we don't know. Cilia move, and there are these sensory cilia that can sense the extracellular environment, transmit a signal to the cell, and which manifests as degradation of DAN5 mRNA. But beyond that, there's a lot of questions about how the signal is propagated and what leads to these early molecular asymmetries. But there's a lot of genes required to do those first steps. And not surprisingly, those are the genes that we've identified in a lot of laterality spectrum congenital heart disease. Genes required for making the left-right organizer, genes required for ciliary motility, like the dynein heavy chains, the dynein assembly factors, a lot of the genes that were described at the sessions earlier this morning. And when you look at a large number of patients, and this is 5,400 congenital heart disease patients investigated by exome sequencing, and look at genes that are required for cilia function, they are much, much enriched in heterotaxy patients, as seen here in the red circle. However, they're also enriched in the overall congenital heart disease population. And these aren't just heterotaxy patients, the ones in the red circle, that's my broad laterality spectrum abnormalities. So cilia genes, left-right organizer genes required for at least some percentage of patients with heart laterality abnormalities. Interestingly, we've recently found that a subset of those cilia genes are also required for early valve development. So this was a postdoc and a student in the lab who studied cilia within the heart. So now we've moved from the left-right organizer to the heart itself. And what you see here is a zebrafish heart beating. The little green dots are cilia. They're in the heart. They're found in the forming atrioventricular cushion in a very temporally and spatially dynamic manner. And when you get rid of them, you can't cellularize the AV cushions, which for a lot of the cardiologists here should make you think really hard, because quite a few of these patients have AV valve abnormalities. And so the question we're raising is that there's an overlap between left-right development and AV valve development that is not necessarily just due to the abnormal position of the AV valve, but due to the actual developmental structural abnormalities that are mediated by not having cilia. If you then look at all congenital heart disease, you find that recessive congenital heart disease, which is over here, is very enriched in abnormalities in cilia genes. And this is, again, not looking just at heterotaxy, but all comers, but that cluster is likely very enriched for patients with these laterality-associated defects. So I think the key thing here when we think about all this is that this is more complicated than we initially thought. It's not a linear trajectory from one to the other. And many phenotypes that you see in your cardiology care are maybe related to heterotaxy in a mechanistic manner. So what's next? Next, you have to signal that from your left-right organizer to the heart. And the pathway that's been most clearly implicated there is nodal signaling. And the cool thing about nodal signaling is that it's able to take a really small signal and auto-amplify it very, very quickly. And this whole process between having a left-right organizer and having a heart actually start to loop is also only about 2 to 3 hours. You have a primitive heart tube in the mouse at about 4 or 5 somites, and by 7 somites, that heart has started to loop, and the process has really been initiated and pushed in its irreversible direction. Each somite is about 1.5 hours of mouse development. So now let's look at nodal signaling. When we again look at this large cohort, and this is 2,300 laterality spectrum congenital heart disease patients, of which 900 were clear heterotaxies, and the others fall into my more broad definition. And we're looking at mutations in the nodal signaling pathway. There's quite a few of them. So these numbers, there's 32 nodal mutations, 29 SMAD mutations, 24 GDF1, and that's really just sort of starting to look at it. But what I also found interesting when I took this apart is the nodal signaling pathway is outlined on the left, and you would think that if you mess up nodal and its obligate partner, GDF1, you would get a similar cardiac and laterality phenotype. And the answer was really not, it wasn't as perfect as I thought it was going to be. And then if you look at nodal and its downstream signaling components, SMAD2 and 4, the phenotypes again were significantly different. And the most striking things was that nodal mutations seem to be most strongly associated with cardiac effects and not abdominal and other situs abnormalities. So a lot of the nodal patients were isolated L-loop, so the corrected transposition patients. And if you compare that, the blue is cilia defects, and you can see that they are quite different. The cilia defects were not as enriched in the detransposition, and they were remarkably enriched in the atrioventricular canal defects, which suggests to me that there might be something going on with the AV valves, which is something that from a cardiology perspective becomes very important, that we figure out which genetic defect has which cardiac effect, which may predispose the patients to certain types of outcomes. But this is really, this was kind of work that I did for the last week preparing for this conference, so this is really not ready for prime time, and we're growing the numbers of patients. This is only looking at single nucleotide variants. This is not yet looking at splice site variants, structural variants, so this is going to be a much more granular data set as it gets ground out over the next couple of months. So what are the clinical implications? And those have already been hinted at. The genetics impacts the outcomes. So again, when you look at a large cohort and you look at whether they have cilia gene defects, other gene defects, and you start associating them with particular phenotypes, the greatest odds ratio for affecting outcomes was having a combination of a heterotaxic heart, a cilia defect, and those two together increased your risk of postoperative mortality and increased your risk most significantly for prolonged ventilatory requirement postoperatively, which based on this morning's presentations is not totally shocking, because if they can't clear airways and you put an endotracheal tube in, you are buying yourself some problems. So these are damaging variants with increased arrest, and damaging variants in cilia genes increased risk of greater than 7 days of postoperative ventilation. So the clinical implications extend beyond the immediate surgical effects, but also that cilia are in lots of organ systems. They're in your kidney, they're in your brain. They're used to build limbs during development. So I don't like to think of all of this as a birth defect, because that implies, first of all, a defective person. And it implies a fixed thing that may or may not be able to be fixed. I see these genetic variants as a disease that a patient has that can manifest as a structural heart defect, which we've actually gotten, as mentioned earlier, phenomenally better at fixing. We've become better plumbers over time. But what we're left with is patients that have lifelong morbidities, and these lifelong morbidities are turning out to have really huge impacts on their quality of life. So identifying what the genetic cause of the problem is may help us modify the pulmonary disease, renal dysfunction. There may be association with obesity. Cilia are in your brain, and they're required to regulate appetite. So I think we should start looking. Some cilia variants are associated with visual defects, hearing problems. So these are all lifelong morbidities that as primary caretakers we need to be aware of to work with the patient and their family and take care of not just the heart, but the whole patient. So I like to think of this as cilia-associated heart disease as opposed to some sort of congenital heart defect. So the clinical implications beyond that are obviously family counseling. These variants have a significant familial recurrence rate. That depends on exactly what the variant is. The phenotypes are incredibly variable, and there's a lower rate of parent-offspring transmission in heterotaxy and laterality-associated disease than in other congenital heart disease, because a lot of these are recessive as opposed to dominant. And I think I'd like to acknowledge a lot of the people that helped with all of this work, specifically all the patients and families that contributed to the genetic study that now encompasses 17,000 congenital heart disease patients, of which over 2,000 have some sort of a laterality-associated disease. And we are going to be able to follow them longitudinally if the NIH blesses it, and see how the genetic variants translate into outcome risks, hopefully some of which are modifiable. And then a whole lot of the people that worked with us on both the consortium and the mouse work. Thank you.